Good day. This is the Professor, CBMD PhD. And today's Tuesday, April 4th, 2023. It is 2:45 and 14 seconds, a.m. Eastern Standard Time. And today's research, neutrophils. So let's hop to it here. A surprise finding shows that neutrophils can be key anti-tumor weapons. Date March 30th, 2023. Source: Weill Cornell Medicine. As white blood cells called neutrophils have an unappreciated role in eradicating solid tumors, according to a surprise discovery. Researchers investigated how a T-cell-based immunotherapy was able to destroy melanoma tumors, even though many of the tumor cells lack the markers of antigens targeted by the T-cells. They found that the T-cells, in attacking the tumors, activated a swarm of neutrophils, which in turn killed the tumor cells that the T-cells couldn't eliminate. The findings could lead to new immunotherapies that harness this unexpected but potent anti-tumor immune response. Researchers have tended to think of innate cells as immune cells that at best can help stimulate initial T-cell response to a tumor. In addition, many studies have shown that neutrophils support tumor progression, but here we reveal that they can have a critical role in eliminating and finishing off a tumor that would otherwise escape a T-cell therapy. And this work clearly shows us that the monolithic term, neutrophil, needs to be more specific, based on the use of single-cell technology. In the past, this general term referred to as a population of cells that were not thought to be helpful in controlling tumors. We now know that a subset of these cells is critical in optimizing engineered T-cell therapies to overcome heterogeneity. As cancer immunotherapies, which harness or boost immune cells' ability to attack malignant cells, have begun to revolutionize cancer treatment over the past decade. In some cases, these therapies have essentially cured advanced cancer patients who would have had no hope of survival otherwise. Yet, for most cancers, immunotherapies are effective in only a minority of patients. In general, researchers still have much to learn about how anti-cancer immunotherapies work and how their effectiveness can be improved. In the study, the researchers investigated an experimental immunotherapy that includes a drug to boost T-cell activity and proliferation, plus T-cells that have been engineered to recognize a melanoma-associated antigen. As tumors sometimes can evade an immunotherapy targeting a specific tumor antigen simply by ceasing to express that antigen, the tumor cells that don't express the antigen are called escape variants. However, the researchers found that the boosted T-cell therapy could eliminate melanomas in standard mouse models, even when a large portion of the melanoma cells lacked the targeted antigen, TRP1. Ultimately, they determined that the initial anti-tumor activity of the T-cells against TRP1 expressing melanoma cells triggered a secondary tumor-killing response from neutrophils. As these white blood cells are best known as first responders to infections and wounds, as members of the evolutionary order innate immune system, they do not target specific antigens in the way that T-cells do. Yet the researchers concluded that the neutrophils summoned by their T-cell immunotherapy were indeed responsible for killing off the remaining non-TRP1 expressing melanoma cells, at least in part, by secreting the highly reactive molecule nitric oxide. As part of the study, the researchers identified a characteristic gene expression pattern in the anti-tumor neutrophils and found that, in a widely used database of melanoma patients, the greater presence of this gene expression signature in biopsy tumor samples was associated with better outcomes for patients. The results were especially surprising because prior studies have shown that neutrophils around the tumor often act as allies in the tumor. The tumor co-opts them to help it survive and spread, which they do in part by suppressing other elements of anti-tumor immunity. In any case, the new finding suggests that in the context of a strong immunotherapy that includes engineered T cells targeting tumor antigens and a general boosting of T cell functions, neutrophils can play a significant anti-tumor role, in fact, an essential role in mopping up escape variant tumor cells that would otherwise keep the tumor alive. As conventional T cell based therapies have been successful in treating cancers, but they are not as effective against heterogeneous tumors, which have antigenic escape variants that can be invisible to the immune system. It was incredibly surprising to discover that T cell educated neutrophils can eliminate these invisible tumor cells. This discovery highlights the importance of mobilizing multiple arms of the immune system in the fight against cancer. The researchers now are continuing to study these anti-tumor neutrophils to determine how they can best be induced and directed as cancer fighters on their own or as enhancers of other immunotherapies. Researchers also hope that they can find a way to use measures of these anti-tumor neutrophils and tumor biopsies as biomarkers that help us choose the best therapies for patients. And this is the professor of microbiology, bioinformatics, and pathophysiology, leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.